Bismillahirrahmanirrahim. Hello and welcome back, friends. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about sporogenesis or sporulation, or we can also call that endospore formation. Okay, or we also call that life cycle of bacillus megaterium. In the previous video lecture, if you guys remember, we talked about the endospore structure. Endospore formation means endospore layers. And we also talked about the endospores, present in a mother cell, that how endospores are, the position of the endospores inside the mother cells. Okay. Now, in this video lecture, what we are going to talk about? We are going to talk about endospore formation. So let me write endospore formation. Or we can say or a life cycle of bacillus megaterium. We also called this, this process, we also called it sporogenesis or sporulation. So let me write sporogenesis or sporulation. Now, before explaining this topic, means endospore formation, you need to understand about the endospore. Okay, endospore structure. If you missed that video lecture, so I will give the link in the description box, then you will check it. Now, endospore formation, or we can say sporulation or sporogenesis, it is consists of 7 steps. How many steps? It is consists of the 7th step. First of all, what actually happened? We have a mother cell, let's suppose. So it is start from the mother cell. So first of all, we have what? We have this mother cell. Now, this mother cell, it is ready to divide. This is what? This is ready to divide. At that side, we have what? We have the genome. This is also the genome. Now, this is what? This is the cell membrane. And we call that what? We call that mother cell. Let's suppose this is the mother cell. We also call that parent cell, those cells which are ready to divide and produce 2 daughter cell. Now, from the mother cell, what we get? We get 2 daughter cell, but here I will write 1 daughter. Okay, so from the mother cell, what we get? We get 2 daughter cells. So let me write, means here we will write the 1 daughter cell. So this is what? This is the daughter cell. Okay, this daughter cells have the cell membrane also. Now, this is what? This is the cell membrane, and it also contain the genome. Now, in the first stage of this endospore formation, or we can say bacillus megaterium, let's suppose what actually happened inside the environment. Let's suppose this is bacillus megaterium bacteria. This bacteria is present in that environment where there is a shortage of nutrients or moisture. Then such bacillus megaterium start their life cycle, or we can say they are starting to produce endospores. So, in the first stage, what actually happen? They will produce axial filament, or they can form axial filament. So, in the first step, what actually we have? We have the axial filament formation. So, let me write axial filament formation. Okay, in axial filament formation, what actually happen? This cell membrane are closer. This cell membrane means this some area of the cell membrane are get closer to this genome. As you are watching, means this are closer. It is closer to what? It is closer to DNA genome. Now this membrane are closer to DNA means we can say genome of this bacillus megaterium. Once it is close to this genome, then we call that axial filament formation. So this is the first step. After that, what we have, septum is formed, or we can also call that 4 spores development occur. So after that, what we get spore 4 development. So let me draw. So in the next step, what we have spore 4 development. So let's suppose again this is the bacterial cell, means bacillus megaterium, and what actually happened, we get this is the spore 4, 4 spore. This is the 4 spores having the genome. At that side, what we have this structure. So we have this structure. Now this is the septum. As you are watching, we get the septum, and as well as what we get this 4 spores. So let me write, this is the second step. So in second step, what we get in the second step, we get 4 spore development. Or septum is formed. After that, what actually happened in the next step? After that, what actually happened? In the next step, this means this spore 4 are engulfed by this membrane. This membrane is engulfed this 4 spore. So the next step, we have engulftion of the spore 4. So let me draw how. So let's suppose this is again the cell, means bacillus megaterium again, and this is the spore 4, and it is engulfing what, it is ready to engulf this spore 4. Now this is the spore 4 which is engulfed, and these are the genome of the bacillus megaterium. Once so let me write, this is the third step in which the engulftion of spore 4. So let me write engulftion of spore 4. 4 spore. 4 spore. Okay. So this spore 4, this 4 spores are engulfed by this membrane. After that, what we get the cortex. So in the next steps, what we get the cortex. So how the cortex will be look like? So let's suppose this is again a bacterial cell, means this is again the bacillus megaterium. So inside the bacillus megaterium, this is the one, and this is the second one. Okay, and this is called spore court. What we call that? We call that a spore court, and this is the cell membrane area. Okay. So this is called what? This is called cortex. So let me write. Okay, so in the fourth step, what we get? In the fourth step, the cortex are formed. So let me write cortex formation. Now let me a little bit explain this. What actually happened in the fourth step? The cortex is formed, and this cortex is present between the two membrane. As you are watching, this is the first one membrane, and this is the second. Okay, so this is what? This is the first, and this is the second. So we get the cortex between these two membrane. Okay, not only that, calcium and diploclonic acid is also formed at this stage also. After that, what we have? We have another step occur, and in these steps, what actually happen? The protein coat are formed. So in protein coat, how it will be look like? So in this case, the cell will be look like in this form. So let me draw. Again, this is the cell, means bacillus megaterium, and at that stage, the bacillus megaterium can form what? They can form protein coat. So let me draw. So this is, let's suppose what? This is, let's suppose, the cortex area. This red one, let's suppose, represents the protein coat. Okay, so what we get? We get the protein coat. So after that, what we get? We get the protein coat. So let me write, this is the fifth step. So in fifth step, what we get? We get the coat formation. Coat formation. Now in the next step, what actually happen? Now in the next step, we get another layer around this coat, means around this protein coat, we call that exosporium. So let me draw again the bacillus megaterium. So in the next step, what we get? We get the, let's suppose this is again the bacillus megaterium, and this is the membrane portion. Okay, and this is what? This is the first layer. Okay, sorry. Let's suppose this is what? This is the cortex. Here are the genome, and after that, what we have? We have the, spore coat. Coat, and then at the end, what we have? We have this like structure. We call that exosporium. So this is the another layer which we call that exosporium. Okay, so in this steps, so this is the 6 steps. In the 6 steps, what we get? We get exosporiums. Exosporium formation. Once we get exosporium, then at the end, what we get at the end? This, this bacterial cell, or we can say this bacillus megaterium, can produce lactic acid enzyme, or we can say they can produce lactic acid, or they can produce the enzymes. Due to this enzyme, what actually happen? These, these cell membrane and the cell wall are ruptured. So the cell membrane and the cell wall are what? The cell membrane and the cell wall are ruptured, and finally what we get? We get only spores. What we get? We get only spores. So let me draw again a bacterial cell. So this is again a bacterial cell. Sorry, not at this stage. This is not a bacterial cell. We can only get this spore. So let me draw. So first of all, what we have? We have this means exosporium. After the exosporium, what we have? We have this protein coat. After that, what we have? We have the protein coat. And after the protein coat, what we have? We have this cortex. Okay, and inside the cortex we have the genome. Okay, so that's how the process is completed, and again this process is started. So this is the final steps, and this steps is we can say that in the 7th step, what we get? We get a complete spore. So this is the complete spores. Complete spore. Okay, so that's all about the life cycle of bacillus megaterium. Okay, so that's all about what? That's all about the endospores formation, or we can say the life cycle of bacillus, or we can also call that sporulation, sporogenesis. Okay, what actually happened inside this bacillus megaterium? This process required 10 hours. How much? They can require almost 10 hours. They required 10 hours to start the process or to complete the process. So I hope you understand about that video lecture. If you like this video, so make sure to hit the like button, share this video, and if there is some point missing, so then write it into a comment. Inshallah, I will explain it. So like the video, share the video, and don't forget to subscribe the channel. Subscribe the channel to get more interesting videos like that. Thank you. Take care. Bye.